Hi, my name is Gustavo, and I would like to tell you about my method for pathway analysis that uses mutual information to score the relationship between pathways and phenotypes. So first, a very brief introduction of why we're using mutual information to do this. Essentially, mutual information is a measure of dependence between two different variables, but it's one that's extremely flexible. So there are many ways to mathematically define mutual information, but I find this one the most intuitive, which is the KL divergence between the joint distribution of distributions variables x and y and the product of the marginal distributions. And as a visualization here, we have two different distributions of points, and you will see that both of them have exactly the same marginals. But of course, the joint distribution is very different. Here, points, the x and y variables are mostly independent. That means that this joint distribution of points is almost the same as the product of the marginal distributions. So this will have a very low mutual information between variable x and y. Here, of course, the picture is much different. And you can see that here the variables are very much interlinked. And this is what mutual information is measuring, the difference between the distribution on the left and the distribution on the right. And of course, it's important to see here that we didn't need to make any assumptions about the nature of the relationship between these two variables. All we need to have a mutual information value is the marginals and the joint distribution. And with that, we can move on. So now, how do we use mutual information to do pathway analysis? Well, this is our idea. We start with gene expression data. So for each pathway, we select the genes belonging to that pathway and subset the gene expression data. And for each pathway, we generate a nearest neighbor graph. And again, I just want to emphasize that to calculate the neighborhood of each sample, we only use a distance that's calculated only by the gene expression of the genes inside that pathway. So we do this separately for each pathway, which means that in the end, we have one graph for each pathway. But it's important to note that each graph has the same number of samples and the same number of edges. They're just wired differently. So now for each of these graphs separately, we do a module detection step. Here we use the Leiden algorithm, which is a greedy algorithm that maximizes the modularity of the graph. But importantly, as a result, we have the samples in this pathway separated into modules. Now we have a module assignment for each sample and a phenotype that also associated with each sample. And with these two variables, we can calculate the mutual information. But importantly, we also can calculate the maximum mutual information that has to do with the entropy of these two distributions of samples. And also, we can calculate the expected mutual information if the assignment of the samples were random. And these we calculate because we want to now have a pathway score that goes from 0, which will be the expected mutual information at random, to 1, which will be the maximum mutual information that we can achieve using these two distributions. And this makes a score that goes from 0 to 1, and that can be compared across multiple experiments. So now a brief intuition of the signal we are trying to capture. We applied our method to a single cell dataset, scoring the pathways against the cell types in the dataset. You can see there are all here UMaps, and in each UMap, it was constructed only using the genes of a specific pathway. So first, let's look at pathway A here. This is a very high scoring path using our method. And you can see that indeed, using only the genes on that pathway, we can separate the cell types very well. Pathway C and D have lower scores, And you can see that with the lowering scores, you see a more homogeneous distribution of cell types in the data set. And finally, we look at path B, or pane B, and we can see that here, instead of coloring it by cell type, we color it by which part of the body the samples were taken, either the bone marrow or the liver. And you can see that although pathway A separates very well the cell types, it does not separate the samples by which part of the body they were taken. In summary, some advantages and disadvantages of our method. Well, it captures nonlinear interactions, it does not rely on gene-level statistical tests, and it has only one parameter, which is the number of neighbors we use for the graph. And that parameter doesn't have a big influence on the final results. The test is self-contained. That means that to score a pathway, we only need to look at the genes of that pathway. Also, mutual information is very sensitive, so we can capture with high confidence even small effects. And it also provides a natural way to deal with confounders, which is by the use of conditional distributions instead of the full joints and marginals. And some of the advantages is that it only scores discrete variables, because in the end we need an empirical distribution. So you can use it for continuous variables as well, but you have to discretize them in some way, either by through a histogram or some other sort of quantization. And it needs more data points than other established methods. With this, I'd like to thank you for listening to my presentation. If you enjoy our projects, we have a preprint and code available at those following links or the QR code, and I'm also open to questions.